Good morning, everyone, and thank you for being here today. I am delighted to welcome you to this presentation. My name is Dr. S.K. Singh, and I hold a PhD in pharmaceutical sciences. Today, I will be discussing an important topic, the incorporation of recovery processes in oral solid dosage forms, or OSD, and how these processes can impact their shelf life. Over the course of this session, we'll explore the key concepts behind recovery methods, their practical applications, and the significance of optimizing shelf stability. So, let's dive in and explore this fascinating subject together. Let's start by talking about oral solid dosage forms, or OSD, which include tablets, capsules, dry syrups, and sachets. These are common forms of medication that people take daily. Now, in the manufacturing process, we often encounter situations where we need to recover materials. This recovery can involve reprocessing, reworking, or simply reclaiming materials that didn't meet quality standards initially. Some typical sources of these recoveries include blend leftovers, which are the excess powders remaining after mixing, tablet rejects caused by compression defects, and coating rejects, which happen when the coating doesn't meet quality requirements. It's important to understand that reprocessing isn't just about saving materials. It also raises regulatory concerns. Specifically, we need to consider how these recovery processes might impact the overall quality of the product and its shelf life. Maintaining consistent quality and stability is critical, so while recovery is useful, it must be carefully managed to ensure we don't compromise the effectiveness or safety of the medication. Let's start by looking at the definitions on this slide. Recovery or reprocessing refers to the reintroduction of materials back into the same process stage. Essentially, when a material doesn't meet specifications initially, we can sometimes reintroduce it into the process to optimize resources. On the other hand, reworking involves an additional processing step beyond the original process. This means that if a product or material requires further processing to meet quality standards, we perform that extra step to ensure it complies. Now, it's important to emphasize that, according to good manufacturing practice, or GMP, any recovery or reworking activities must be properly justified, validated, and thoroughly documented. This ensures that these processes are controlled and consistent, maintaining product quality and patient safety. Connecting this to broader quality frameworks, these activities align with key guidelines like ICH-Q8, which focuses on quality by design, ICH-Q9 on risk management, and ICH-Q10 regarding the pharmaceutical quality system. All these guidelines reinforce that any reprocessing or reworking must be scientifically justified and systematically managed to ensure continuous quality. Let's take a look at the different types of recovery in OSD. First, we have granule recovery, which involves reclaiming granules during manufacturing or processing. Then there's tablet crushing and regranulation, a process used to reuse crushed tablets by reforming them into usable granules. Next is blend reuse, where leftover or unused blend materials are recovered for future use. Coating recovery is also possible, but it's limited because coatings can be sensitive and might degrade over time. Lastly, dust collection reuse can be done, but it carries a high risk since dust may contain contaminants or unprocessed material. Now, how do these recovery methods impact the critical quality attributes, or CQAs? Well, they can affect several key parameters. For example, potency or assay results can be altered if the recovered material isn't identical to the original. Dissolution rates may change if the recovery process affects the drug's release characteristics. Degradation products can increase if the material is exposed to conditions that promote instability. Moisture content can also be influenced, which in turn impacts stability and shelf life. Hardness and friability are important too. If the recovered material affects the physical strength of the tablet, it could lead to breakage or compromised integrity. It's important to remember that ICH guidelines require us to monitor these CQAs throughout stability testing. By doing so, we ensure the safety, efficacy, and quality of the final product are maintained, no matter what recovery processes are involved. Let's take a closer look at the regulatory expectations outlined in the ICH-Q1 stability framework. First, it's important to understand that stability testing must accurately reflect the final commercial process. This means the conditions and procedures used during testing should mirror those used in manufacturing and packaging for the product's market release. Next, we need to ensure compliance with GMP requirements, which are essential to maintain product quality and consistency. These guidelines help us ensure that the stability data we generate is reliable and credible for regulatory submissions. Another key point is that stability studies should not have an adverse impact on critical quality attributes, such as assay, dissolution, and impurity profiles. In other words, the stability conditions should not cause any degradation or changes that could compromise the product's safety, efficacy, or quality. Finally, regulatory expectations specify that a minimum of 3 batch stability data points are required. This helps demonstrate the batch-to-batch consistency and robustness of the product over time. Overall, these points reinforce the importance of comprehensive, consistent, and compliant stability data to support product approval and ongoing quality assurance. Let's take a closer look at the stability study design based on ICH Q1A guidelines. When we evaluate stability under typical conditions, we need to consider several key factors. First, risk factors stemming from recovery processes can influence stability. For example, increased moisture exposure can accelerate degradation or cause physical changes in the product. Mechanical stress, such as vibration or handling, can also impact stability by inducing physical alterations. API degradation is another major concern, as active pharmaceutical ingredients can break down over time under certain conditions. Variability in parameters like particle size distribution and blend uniformity can also affect stability, since inconsistent mixing or particle sizes can lead to uneven degradation or performance issues. We also need to be aware of possible polymorphic changes, which are different crystal forms of the API that can affect solubility and bioavailability. Additionally, increased impurities might develop over time, potentially impacting both safety and efficacy. Overall, understanding these factors helps us design more robust stability studies and ensures that our products remain safe and effective throughout their shelf life. Let's take a closer look at the example of dissolution impact. Without recovery, we're seeing about a 95% drug release at 30 minutes. However, if recovery is included, that percentage drops slightly to 88%. This highlights how recovery can influence dissolution results. From a regulatory perspective, dissolution failure is considered a significant change. According to ICH guidelines, such failures can trigger additional studies or even rejection of the product. It's important to understand that even small differences in dissolution profiles can be critical. They help us assess product consistency and ensure quality throughout the development and approval process. So, keeping an eye on these parameters is key to maintaining regulatory compliance and ultimately delivering safe, effective medicines. Let's take a closer look at shelf life estimation, based on the ICH Q1E guideline. This part of the process is really critical because it helps us determine how long a product remains effective and safe to use under specified storage conditions. The key point here is that shelf life isn't just a guess. It's scientifically derived using data from stability studies. We analyze how the drug degrades over time, considering factors like temperature, humidity, and light exposure. ICH Q1E provides a structured approach for this assessment, emphasizing the importance of designing stability studies that are relevant and comprehensive. This way, we can model how the product behaves over its intended shelf life accurately. Importantly, these estimations support our overall goal of ensuring product quality and patient safety. When we establish a reliable shelf life, we're not just meeting regulatory requirements, we're building trust with healthcare providers and consumers. So, in essence, Shelf life estimation is about using scientific data to predict how long our product stays within its quality specifications, helping us make informed decisions about packaging, storage, and labeling throughout the product's life cycle. When we look at regulatory risks, there are several key issues to keep in mind. First, batch rejection can occur if the manufacturing process doesn't meet the required standards, which can delay approvals or lead to product recalls. Shelf life reduction is another concern. If stability studies show the product degrades faster than expected, it can impact how we plan for distribution and inventory. To address these risks, it's often necessary to conduct revalidation, perform additional stability studies, and if there are any changes after approval, we need to submit post-approval change filings or variations to regulatory authorities. Now, moving on to the statistical considerations. It's important that we analyze data from at least 3 batches to ensure consistency and reliability. We typically examine stability at multiple time points, such as 0, 3, 6, 9, and 12 months, to get a comprehensive picture of how the product performs over time. Regression analysis is a key tool here, whether linear or nonlinear, so we can model the stability data accurately. And finally, when assigning the shelf life, we aim for a 95% confidence interval. This level of confidence helps ensure that the shelf life we determine truly reflects the product's stability profile, giving both manufacturers and regulators the assurance needed for safe and effective use. Let's talk about some key mitigation strategies to manage risks effectively. First, it's important to limit the recovery percentage, aiming for something like 10 to 20% or less. This helps minimize potential impact if issues arise. Next, adopting a risk-based approach, often guided by quality by design principles, allows us to proactively identify and control critical factors throughout the development process. We also need to conduct thorough stability studies, including comparative stability testing, to see how our product behaves over time, and forced degradation studies to understand how it responds to extreme conditions. These give us valuable insights into the robustness of our formulation. On the control side, controlling moisture levels is crucial because moisture can significantly affect stability. Managing particle size is also important, as it impacts the product's uniformity and performance. Lastly, ensuring consistent mixing uniformity helps maintain the quality and efficacy of the product. By integrating these strategies, we can better safeguard our product quality and ensure a successful development process aligned with our overall risk management goals. Let's take a look at some practical industry limits, which are widely accepted practices in the field. Although these aren't legally fixed, they serve as useful benchmarks for evaluating risk and stability. Typically, the acceptable range for impurities or variability is categorized as follows. If the level is less than or equal to 5%, that's considered very low risk. In most cases, this is acceptable without raising major concerns. When the level falls between 5 and 10%, it's generally acceptable, but only if you have proper justification and validation to support that. For levels between 10 and 20%, you'll need strong scientific justification and comprehensive stability data to demonstrate safety and efficacy. And finally, if the impurity exceeds 20%, that's considered high regulatory risk. Such levels are rarely accepted and often require significant justification or reformulation. These guidelines help us navigate industry standards and regulatory expectations while ensuring product quality and safety. Let's take a closer look at a practical example of scientific justification. Here, we see how a well-supported rationale is built around empirical evidence and logical reasoning. It's crucial because it shows that our approach isn't just based on assumptions, but on solid scientific principles. When we develop these justifications, we're essentially connecting our objectives to existing knowledge, demonstrating why our methods are valid and necessary. This step is fundamental in convincing stakeholders and ensuring the credibility of our work. Remember, a strong scientific justification ties everything together and reinforces the overall integrity of our project. Let's wrap up with some key takeaways. Recovery processes, when used, should always be backed by solid scientific justification. They have a direct impact on critical factors like stability and shelf life, which are essential for ensuring product quality and safety. It's important to remember that any recovery step must comply with established guidelines, specifically ICHQ-1A for stability and ICHQ-1E for evaluation. Additionally, adherence to GMP and QBD principles is crucial to maintain control and consistency. If recovery processes are poorly controlled, it can lead to a shortened product life cycle and increased regulatory risks. So, maintaining strict control and proper justification isn't just best practice. It's essential for protecting both the product and the regulatory compliance. Always keep in mind that careful, scientifically justified recovery procedures support the overall integrity and success of our pharmaceutical products. For further support, please feel free to contact us. We're here to help you with any questions or additional information you might need. And with that, I want to sincerely thank you all for your time and attention today. Your engagement is what makes these discussions meaningful. Remember, we're always available to assist you in applying what you've learned and moving forward confidently. Thank you again.